So first announcements, there'll be grad student postdoc lunch in McCrary after this. And there are still dinner slots open, I think 2. So if you want to come to dinner, let me know. So I'm really excited to introduce Dr. Amy Iler, who is a conservation scientist at the Chicago Botanic Garden, as well as a professor at Northwestern University. So Amy got her PhD at Ohio State and then went on to do her postdoc with David Inouye at the University of Maryland in the Rocky Mountain Biological Station. Before she moved to Denmark as a Marie Curie Fellow, which sounds like a super cool fellowship because basically they pulled together a bunch of scientists in different fields. She was telling me there's historians and musicians and like a whole bunch of people who have to work together. And so she's going to tell us a little bit about a collaboration that came out of a fellow biologist interaction, so that's kind of cool. In 2016, she joined the Chicago Botanic Garden, and her lab focuses on how variation among species affect both population dynamics and interactions. So she's focused a lot on flowering phrenology and on how that influences species success and pollinator interactions. She also, because she's in the CPG, does a lot of outreach efforts, particularly with pollinators and climate change, and runs the Plants of Concern program, which monitors rare plant populations across Illinois. And so I'm going to hand it over to Amy.
Thank you, Meredith.
Hi, Sean. Hi, everyone. I'm really excited to be here. I've always wanted to visit Kellogg, so I finally get to see it. As you'll see, I'm a field station person too, because I work at the Rocky Mountain Biological Lab. So yeah, so today I'm going to tell you about some of the research. A really big focus in my lab right now is trying to understand the ecological consequences of shifted flowering times under climate change. So we know climate change is happening and it's having all sorts of biological effects across the globe. And we can group these ecological responses to climate change into some main categories. So we are seeing species moving through space to try to track their preferred environments. Typically we see species moving up in elevation or up in latitude. Also, species are moving through time to try to track preferred environments. On average, species are shifting earlier in time as the climate changes. And we're also seeing, species might stay and adapt to their new environments with some trait changes. And in insects, we're seeing a lot of insects reducing their body size and under warmer temperatures. And then finally, we might see some extinctions. So those are some main responses, and this is what I focus on, not surprisingly, shifts in phenology in response to climate change. And I want to mention too, these are not mutually exclusive. We're seeing species exhibit a range of these responses. So as I mentioned, we see a variety of changes in phenology, but mostly earlier events. So this is a figure from some of my work as a postdoc from a data set in Colorado where we have flowering times for all these species. They're listed in the order in which they flower from early to latest here. And I'm just showing one species right now at the top. And there's peak, first, and last flowering dates. And in this case, they've all gotten earlier. So I'm going to put them all up here. And what I mainly want you to notice is that to the left of this dotted line, those are all earlier flowering events, and then to the right of the dotted line would be later. And there are some species whose last flowering dates are extending later into the season, but you can see most dates are getting earlier, and this is true for pretty much most temperate environments around the world. This is a very common thing. Mostly earlier events, a range of magnitudes and the shifts, but also some later events. So we're getting a pretty good handle on how climate change is affecting phenological events, but we know much less about the ecological consequences of those shifts. And so that's what a lot of my research right now is focusing on. And today I'm going to tell you 3 different stories. So the first one, we're going to ask if earlier flowering under climate change affects population viability in a sunflower. Then we're going to ask if this affects soil microclimates. This is not immediately apparent why this would be true. This is the story that came out of the collaboration with the cell biologists, so I will elaborate more when we get there. And then the third is my recent master's student who finished looking at whether earlier flowering affects interspecific interactions between plant species. So all of this work takes place in Gothic, Colorado. In western Colorado, shown here by the star. And this is at the Rocky Mountain Biological Lab. So this is a field station that is 2,900 meters above sea level, which is 9,500 feet. It's in the subalpine in the Rockies. And this field station was an abandoned silver mining town that was converted into a field station in the early 1920s. And so there's a bunch of old dilapidated cabins, also some nicer newer ones that researchers come stay in. And it's not affiliated with a university like Kellogg. So I have to say I was impressed with the dorm that I'm staying in. It was so nice. I'm like used to these old mining shacks. Although now I've been here for 10 years, I'm in a pretty nice cabin now, I have to say. So yeah, that's the story of the Rocky Mountain Biological Lab. There are a lot of researchers here studying climate change, so it's a really great place to study climate change. You have a lot of people working on similar questions, and there's also a lot of long-term data similar to here. How is the climate changing at the Rocky Mountain Biological Lab? I might say rumble, that's what we call it for short. But we see that the summer temperature is increasing. This is since 1974. And then snowmelt dates, which I'm showing here on the right-hand side, are getting earlier. And there's a lot of variation around this regression line. So although there's this trend toward earlier snowmelt, it's hugely variable. And I want to point out, you'll see some phenological events shown as day of year throughout the talk, and that's just January 1st would be one. And so, for example, this regression line goes from June 1st to May 15th. And we can get a better idea in terms of snowmelt dates, how they're changing over a longer time frame by looking at stream gauge runoff information, which we have going back to the 1920s. And if we look at that, basically what happens is when the snow melts in the mountains, the streams really pick up and they flow very fast. And so using that as a proxy, going back to 1920, we see a significant change in terms of earlier snow melt through time. And this is something we're seeing across the Western United States. Snowmelt dates are getting earlier for two reasons: because it's warmer, so the snow's melting faster, but also we're seeing less snowfall over the winter as well. And the timing of snowmelt is an important ecological factor in these types of ecosystems. So this is a picture. This was taken in May of 2011 when I was there as a postdoc. And this is uncommon to have this much snow in May. But it can happen. And the point is that really, the growing season doesn't start until the snow's gone. So at least for the herbaceous plant community, nothing happens. And then as soon as that snow melts, the first species to flower comes out pretty quickly. It's also the main source of water for this ecosystem. So we get the snowpack as it melts is a huge amount of water, and then it's pretty dry. So the snow melts mid-May, and then it's dry until early July when we get monsoon rains that come through. So one thing we're seeing is as that snow melt date gets earlier, there's a longer dry period. Does that make sense? Cool. Okay, and then we're also seeing the temperature and the timing of snowmelt are strongly associated with flowering phenology. So warmer temperatures and earlier snowmelt dates lead to earlier flowering. So the species I want to talk about is Helianthella quinquenervis, aspen sunflower. It flowers in mid-July, and it's a long-lived perennial. So it's also very common and abundant in these subalpine meadows. So a lot of pollinators visit it, a lot of different herbivore insects are present on it as well. And it is, like most species, sensitive. Its flowering date is sensitive to when the snow melts. So this is just to show you that, indeed, as snow melt dates become earlier, flowering also becomes earlier. Another thing that is, we've learned about this species, and these are data from David Inouye that he's been collecting since 1974. No big deal, right? That the flower abundance is highly variable from one year to the next. And in particular, there are many years when there are no flowers at all. And these are years when snowmelt is earlier than average. So we see this relationship between early snowmelt and low flower abundance. Does anyone know why? I heard you like to interrupt, so I thought I might throw a question out. Competition, that's a good idea. Maybe there's more competition. Drought, so maybe it's dry. When it's drier, they can't make as many flowers. That is the most obvious answer, but it's actually frost damage. So it's a counterintuitive effect of climate change that we're seeing increased frost damage in some species as they flower early. But what's happening is now you've got a plant species that's flowering at an earlier date than it's used to, and it's now exposed to lower nighttime temperatures. Oh, question, yeah.
No, we're supposed to know that here in Michigan, because that's why they grow all those fruit trees along Lake Michigan. It's cool, and they don't flower.
That's right. This is related to fruit trees in Michigan. So in 2012, I think Michigan lost its entire cherry crop because the trees flowered early and they got frosted. Yeah. Yeah, that's happening in Colorado as well. So when that happens, this is what the flower stalks look like. They don't go on to make any seeds or flowers that year. So to show you what this looks like, here's the meadow where we study this plant species. All of the yellow flowers are Helianthella. And this picture was taken in a year when snowmelt was late. And compare that to a picture taken on the same date in a year when snowmelt was early. So it can be a really huge effect. And in some cases, 100% of the flowers are lost to frost. So you might think this should have an effect on the population, but we don't know unless we do a population model. So I really want to focus on this question of how does early snowmelt affect population dynamics in this plant species? So I want to take you through some hypotheses. We've already seen that earlier snowmelt is expected to lead to increased frost damage and decreased reproduction for this species. If we think about vegetative growth, we could see increased growth in response to early snowmelt because there's more time for them to grow. The growing seasons are longer. Alternatively, as someone brought up already, it could be drought. So earlier snowmelt could lead to the opposite, decreased growth, because there might be water stress during that time. And finally, for survival, these are long-lived perennials. So I actually wasn't expecting to see a huge effect of snowmelt on survival, but we could see decreased survival because of the drought period as well. And then also, I want to jump back to reproduction. There could be a direct effect of early snowmelt on reproduction via drought effects, which is what was mentioned earlier as well from the person in the back. Alternatively, we could see increased reproduction. And this could be a compensatory response. So something we see a lot in long-lived perennial plants is that if they can reallocate resources across years. So if they take a hit in one year, they might be able to allocate more resources in the following year. So this would be a lagged response, if anything. And this is the same idea as like a cost of reproduction. If you reproduce a lot in one year, you might see lower reproduction the year after. So to get at this question, we used a long-term demography study that was started in 1998. And this is also started by David Inouye, who was my postdoc advisor. And we have all of these tagged individuals. So every individual has a unique tag. And we go to them all every year and measure their size, which in this case is just the number of rosettes or above ground meristems. We count those. And we keep track of the number of flowering stalks that are frosted and the number of flowering stalks that are okay and go on to make flowers. And we've actually have this type of information for 3 different populations. One at the lowest elevation is at 2,700 meters, so it's around 8,000 feet above sea level. The one in the middle is at the Rocky Mountain Biological Lab, and then there's one higher up that's just about 11,000 feet above sea level. And this represents the elevation range of this species, where we tend to find it. So in addition to this wonderful long-term demography study, I did a snow removal experiment in 2014. So I skied out to the field station early, which I had to do anyway, because at the time I was doing, I was collecting the long-term flowering data, and you have to get there before they start to flower. So I was there anyway. I thought, oh, might as well shovel some snow. So this was a lot of work, and I really have to thank my collaborator, Paul Caradonna, who, despite my best efforts, ended up removing more snow than I did. So we dug the snows out when it was like a meter deep, and we find that leads to about 10 days to 2 weeks earlier snow melt, give or take, in the system. So here's what the plots looked like as they were melting out. So I collected all the same demographic data in 2014 and 2015, the year after the snow melted early. So what I did was I put all the long-term data into a integral projection model. So if you've heard of a matrix population model, it's the same idea. So in a matrix model, you have a probability of transitioning from one size class to the next. In an IPM, you take advantage of continuous size distribution. So all of the vital rates are modeled by linear regressions. And then you get what's called a projection kernel instead of a projection matrix, representing all of the possible transitions. And you end up integrating over this, this distribution. So technically it's a discrete approximation of a continuous variable, which is why I'm showing all these little tiny bars here. But the idea is that you have a lot more power because you're utilizing continuous variation. And so you've got, state X individuals surviving and growing to state Y, and then the reproduction component of state X individuals reproducing to state Y as well. And you integrate to project from one time step to the next. So that's why it's called an integral projection model. So I want to show you all of the individual vital rates. The first one is frost damage. It's not size dependent, so it doesn't matter what size you are, you're equally likely to get damaged by frost. So we've got our 3 elevations, and I'm showing snow melt date here on the x-axis and the probability of abortion in response to frost damage, because as I showed you, that whole flowering stalk just gets aborted. It doesn't go on to make fruits or seeds. And we find, before we didn't know if this would happen at all the elevations, but we did certainly expect to see it at mid-elevation, and we did find the same pattern across all the populations. So that's good. And then also, I found a much higher probability of abortion in response to frost damage in the snow removal compared to the control plots. So next, I want to show you vegetative growth. And for the rest of the vital rates, they are size dependent. So we're going to have plant size here on the x-axis, and it's been log transformed, so it goes from 0 to 4 or 5. And then here we've got plant size in the following year. Excuse me. And then I'm also going to show you how snowmelt date is affecting the vital rate. And I want to mention that for all the vital rates, we actually found a lagged response to snowmelt. So snowmelt the year before affected the vital rate in the next year. Okay, so here is growth. We see larger plants exhibiting more growth, as you would expect, and the effect of snowmelt differed across the population. So what we found is that in the low and mid elevation populations, early snowmelt is associated with increased growth rates, so maybe longer growing season could explain that. And then the opposite, at the high elevation, we're seeing decreased growth under early snowmelt. Maybe a drought response. We're not actually sure why this differed in this case. Okay. And then, I don't know if I said this, the snow removal experiment I did at one location, at the Rocky Mountain Biological Lab. So this is all for, that corresponds to the mid-elevation population. And it's the same pattern, increased growth in this case in the snow removal plots compared to the controls. So next we have plant survival. And there was actually a pretty good decrease in survival under early snowmelt conditions in all 3 populations, but especially in the low and the high elevation population. And we think this is probably due to drought effects or water stress. So I had an REU student look in the snow removal plots, and we found lower soil moisture in the snow removals compared to the controls, and also increased water stress during some parts of the growing season in Healy and Thela. And so for these plants to be scored as dead, we decided they have to be missing for 2 years when we're out there doing the demography survey, because sometimes they get eaten by pocket gophers and the above ground biomass is gone, but they're actually still alive, and so they'll come back the next year. And so I don't have survival data from the snow removal experiment to show you, because I didn't do it for 2 years, but I do have some data from a pilot watering study to show you. And we found consistent results that water, so in this case, we're watering, and watering increases the probability of survival by about 5%. This is not a significant increase. It was only done in 3 plots, but I think it's encouraging that the trend is at least consistent with what we might expect if there was kind of a water stress response happening. Okay, so now we're going to move into the reproductive variables. And what do we see in the observational data? As you would expect, larger plants are more likely to flower. And in this case, this is the compensatory response. So early snowmelt is associated with a higher probability of flowering in the following year. And this was true for all 3 populations. And I saw the same thing in the experiment. So snow removal led to an increased probability of flowering in the following year. Okay, what about the number of flowering stalks that plants make? Larger plants make more flowering stalks, and again, early snowmelt is associated with an increased number of flowering stalks in the year after, in that following year. And conveniently, it's a similar pattern in the snow removal experiment. So a higher probability, or more flowering stalks the year after the snow was removed. So to summarize all of that, this is everything I just showed you in one figure, but the take home is that the observational and experimental results are consistent with one another, which is really nice, and it made me feel better about kind of what the population models could tell us based on this observational data. So what happens if we put all of these vital rate equations into our population model? So I'm going to show you how population growth rate changes according to snowmelt date. So first, we're just going to look at the low elevation population. So what I'm showing here is, again, snowmelt date on the x-axis, and this is a stochastic lambda. It just means that we're able to include some demographic stochasticity into the population model. At 1, we have lambda is steady. So if lambda is 1, the population is neither growing nor shrinking. It's just maintaining. Above 1, it's growing. Below 1, it's shrinking. So a lambda of 0.8 is actually pretty low. That means the populations are replacing 80% of itself every year. So, and that's if it was going to experience this snowmelt date year after year. So that's how to interpret these data. So what do we see for the other populations? We see a similar pattern. The middle elevation population, the decline is not nearly as strong, but there's a slight decrease, and then really decreasing lambda in the high elevation population as snowmelt dates become earlier. Okay, so that's great, but why do we see these responses? So the reason I really like building population models is to get at this question, to see, it's wonderful to have an estimate for how the population might be doing, but then why do we see that? So what part of the life cycle is contributing to these effects? So to do that, I did what's called a life table response experiment. And you basically can understand through which vital rate, in this case, snowmelt is affecting population growth rates. And so again, I'm just going to show you the first one and break it down. So the y-axis here is now the contribution of each vital rate to the response of lambda to snowmelt. So how much does each vital rate contribute to that response to snowmelt? And values around 0 would mean there's a very small contribution. Above means that, in this case, a positive response would be a positive later snowmelt is better and earlier snowmelt is worse for lambda, and then vice versa for values below. So for this one, the take home, this purple line on the top is the contribution of survival. And in this case, snowmelt date affects population growth primarily via survival. That's the biggest one, and it's having a negative effect, in this case, on lambda. I also want to point out the flowering stalk abortion contribution is this red line here, and it is affecting it, but very slightly. And then what do we see for the other populations? A pretty similar picture. You might recall that the survival response wasn't as big in the mid-elevation population, so it's contributing about the same as the frost effect to the decrease in lambda as snowmelt dates become earlier. And then for the high elevation, survival is really having a big effect. So it can be helpful to have these two things plotted on top of each other to see, so you can see how they relate. So here in the middle elevation, the population is not declining as much. And the main reason is because survival isn't as low under early snowmelt conditions as it is in the other populations. And then the reason the high elevation population is really tanking so much as we get beyond mean snowmelt date, which is here, is because you get a negative effect from snowmelt or from survival and from vegetative growth. So if you remember in the high elevation population growth, early snowmelt led to reduced growth, and it was a tiny effect. But this is what's cool, in my opinion, about population dynamics. A really small effect can contribute a lot to an effect on population growth if the population growth rate is sensitive to that vital rate. And in this case, it is. So survival and growth really contribute a lot to the long-term population growth rate, and reproduction not as much. So if we go back to the original question that I proposed for this part of the talk, it was how does early snowmelt affect population dynamics? So we saw that despite a large effect of early snowmelt on flower production, with 100% of the flowers getting lost in some years, early snowmelt mainly has a negative effect on population growth via survival. This frost damage contributes a little bit, but not nearly as much as survival. And I think this is really interesting. It's not actually what I expected to find because it's such a visually striking response and you think, oh, no seeds, that's bad. But actually, if you think about it, what we know about long-lived perennials, it makes a lot of sense because a lot of them are a lot more sensitive to changes in their survival and growth rates than their reproductive success. So that brings me to the second part of the talk. Does earlier flowering under climate change affect soil microclimates? So I'm going to show these two pictures again, where we have our late snowmelt and early snowmelt photo taken on the same date, 2 different years. And this project came out of my time in Denmark, where, as Meredith explained, I was there right before starting my current position. And it was an interdisciplinary institute, and they kind of threw all the fellows together and hoped we would collaborate. So not a lot of oversight. So we decided, oh, let's have a little research symposium early on so we know what everyone works on. And we did this thing called scientific speed dating, which was really fun. So we went around and you had 2 minutes with everyone to just like brainstorm ideas for collaboration. But we also gave little talks. And I was showing this picture, and my colleague Alfonso, this is a cell biologist. In his kind of daily lab routine, he's shooting lasers through cells to try to get them to divide or die, depending on what he's working on. So the point is he thinks a lot about how light and matter interact. So when he saw these 2 photos, he thought, I wonder if when the flowers get frosted, does the albedo of the meadow get reduced so that it actually absorbs more sunlight and it's even warmer and drier? So there could be this like positive feedback where it's warmer and the snow melts earlier and the flowers get frosted and then it's even warmer because the flowers are gone. And I said, that's a great question. I have no idea. So the main idea, albedo is generally, it ranges from 1 to 0. A pure white surface would be 1. I'm showing snow. Snow actually isn't even 1, it's lower, but you get the idea. And then a purely black surface would be 0. So the idea is that because yellow reflects more sunlight than green, the meadows in this condition could be even warmer. So the question is, does frost damage alter the albedo of the meadow and lead to even warmer and drier soils? So we did a field experiment, and I brought Alfonso out to the field station, who's, this is a person who's used to like very sterile lab conditions, lasers and cells. He did great. He did a great job. So yeah, so we removed flowers. We had 2 meter by 2 meter plots. And just removed all the flowers when they're at the bud stage. So we tried to kind of minimize the effects we might have on the plants themselves by not letting them invest fully in flower production and just remove the buds. And this is what it looked like. So the first thing you might notice is these are not squares. These are circles. You'll appreciate this being at a field station yourself. It turns out that another researcher proposed a flower removal experiment on this species in the same meadow as I did. And so we shared plots, naturally. So those plots were larger and circular, and my plots are in the center here. And all of these yellow flowers are the sunflowers. You can see there's some stragglers left. I can assure you they were not in my central plots. I got rid of all of them. So you didn't want to have any flowers in there. But that's kind of what it looks like. And so what we expected then is that the removal plots would have a lower albedo, they'd be warmer, and they would be drier. So the first thing we did, we were able to measure NDVI, which stands for Normalized Differential Vegetation Index, and it's what remote sensing folks use to measure how green the surface of the land is. And so in this case, they were significantly greener. Not a huge effect, but it was a significant one. But does that matter? Stay tuned. So in addition to that, I had an RU student that summer work on measuring getting reflectance values for the flowers and the leaves of the plant. So we used what's called a spectrometer. And here's what this machine looks like. But it has a sample chamber, and you can put something in it, and it will tell you how much light it's reflecting across a range of wavelengths. So we put leaves and flower petals in the chambers, and not surprisingly, flowers reflect a lot more light, as you would expect, than the leaves do. And here's what it looks like over the course of all of the different wavelengths. So this is going from like 250 to 1500 or so nanometers. And at every wavelength, the flowers are reflecting more light. Okay, that's great. But does this differ at the plot level? So do the sunflowers cover enough area of the plot that we would expect to see a difference in albedo or reflectance at the plot level? So what we did was Sarah measured the diameter of a bunch of the sunflowers, and we got an average value, and we estimated based on how many flowers were in each plot, how much light would be reflected based on the coverage. And we did see that the plots with sunflowers should reflect more sunlight. And this is estimated, right? This is an estimate of reflectance. We didn't actually go out and measure reflectance at the plot level. So we're assuming that basically the plots are either green, so here's the value for the leaves, or they're green and yellow. So it's pretty simple, but this is what we found. So the next question is, does this matter for soils? And we buried hobo data loggers 8 centimeters below the soil surface. That's where a lot of the root biomass for Helianthella is located. And we were also interested to see if it affected how stressed out they were. And so that's where we started with our temperature measurements. And what we found is that on sunny days, the soils are warmer in their removal plots. Across all the hours of the day, it's on average 1/4 degree Celsius warmer. So it's a little warmer. It's interesting that this effect was maintained across all hours of the day. And then if we look at just the warmest 2 hours of the day, the plots can be up to 1/2 degree Celsius warmer, which is quite a bit, I think, for that far down, especially. And this is only on sunny days. So if we look at cloudy days, we don't see this effect. So it suggests that it is due to this, the sunlight and reflectance pathway. Okay, so what about soil moisture? We measured soil moisture with a soil moisture meter that you can go stick in the ground, and it tells you the volumetric water content of the soil. And we found that the soils were 1% drier in the removal plots compared to the controls. This is across the 4 weeks of the flowering period. I'm not sure what's going on here. There's no difference in this week. But in all the other weeks, you can see that the removals are drier. There's not as many flowers here during this week. And then if we look pre and post treatment, we find no difference in soil moisture. So that's what we would expect to see if it was in fact something to do with the flowers. So in summary, the results are consistent with this idea that sunflowers could reflect a relevant amount of sunlight and actually affect the soil microclimate. So we found that the plots were greener, they had a lower albedo, they were warmer and drier. Now I will mention, for Sarah's project, she measured water stress in the plants, and they were not more stressed out. So this amount of reduction in soil moisture didn't affect the plants. In the future, I'd like to measure soil respiration because that should be temperature dependent. So we could maybe see an effect on soil respiration. But what we did find just with these data are that flowers could play an important role in phenology-mediated feedbacks between vegetation and climate. So when we think of people that model the land surface of the earth, it's vegetation, it's kind of like urban area, so like non-vegetated area, and soil, and bare soil. And so flowers are usually thrown out, which is understandable. Those are, you know, you can only include so much detail. I think it's interesting that in some cases, flowers might be actually doing something. And of course, this would only be during the time that flowers are there. Mm-hmm.
So earlier you said that areas without flowers had bigger plants. They were apparently growing more biomass, right? At least frost damaged flowers. So
A little bit.
that's why that in these flower removal plots, you might have more transpiration, which would remove more water from the soil as well. Is there a way to control for that in this trend?
Yeah, so the question was, could you see more transpiration when the flowers are present? Oh no, you're thinking when they're absent because the plants are bigger. So I was thinking there would be more transpiration when the flowers are present because flowers are a source of water loss for the plant. And for the growth response, we see that in the year after. So if the plants did grow more, they should be bigger the year following the flower removal. So I think that's a good point. They could be transpiring more. And that'd be something that we could measure in the future. Yeah. Yeah, so actually, where is this work now? It's not published yet. It was rejected recently because we didn't directly measure albedo, because I didn't have $40,000 to buy a piece of the equipment that you need. But I was able to borrow something this summer from another team. They have a field spectrometer, and it's this thing here. It's really amazing. So you can actually, it measures the surface that this little gun is pointing down at. And so I'm in a removal plot here. These are the sunflowers behind me. So I don't have these data processed yet, but I will actually have the land surface albedo and I can compare. Nick?
I'm looking at this picture and it's reminding me that Shunyi, who's in the back, is measuring flower coverage at the scale of 0.7 centimeter pixels using.
Whoa, oh.
You could measure flower cover across all species across the board.
Oh, cool. We should talk, yeah. That's really neat. Okay, so that's the end of that part of the talk. That brings us to the last part, which does earlier flowering under climate change affect interspecific interactions between plants? So I want to bring us back to this figure here, which was showing the variety of species and shifts at the species level, and these can affect community co-flowering patterns. So the degree to which species are overlapping with one another. And we showed this actually by looking at a big matrix of all the interactions. We got our focal species and our interacting species on the top, and once I remove this square, any little green squares will mean an increase in co-flowering, and blue squares are a decrease. And so the main take home is that co-flowering patterns within this community have changed for a lot of pairs of species. So we have some species that are flowering more than they used to, and some that are not flowering or being pulled apart in time. And so now the next obvious question to ask is, what are the consequences of this, if anything? And so we know a lot from some work, that a lot of great pollination biology work that's been done, a lot of it at Rumble actually, but we know that the degree to which plants co-flower with one another can really affect how much they're competing with each other for pollinators and then their subsequent reproductive success. So we would expect to see some changes in reproductive output, maybe due to pollination. But in the context of climate change, there hasn't been any work on this. And it's important because, oh, where'd all my, okay, I didn't know those were layered. The biotic interactions may also be affected by abiotic conditions. So the idea is to take this work and put it in the context of climate change. And so I had a master's student named Marie Faust who did a field experiment last summer at Rumble that I'm going to tell you about now. I'm really excited to talk about her work because it's the first, whoops, this is my first master's student to finish, so I'm really proud of her, and she did great. But her goal was to examine how climate-driven shifts in co-flowering and water availability affect these interactions between two plant species, and then the reproductive success. So what we did was we picked a pair of species that we know to be changing from the long-term data set. The first one is Linum lewisii, or flax, and it's it's the focal species of this study. So this is the one we're going to look at. How does its reproduction change? And the species that it's paired with is Potentilla pulcherrima. And we picked these two species because they're both really common and abundant in the meadows around the field station. So they're pretty easy to work on. Whoops, and this one is, Linum is an obligate outcrosser, so it requires pollinators to make seeds, and it makes more seeds if it gets outcross pollen, whereas Potentilla can self, so we wouldn't expect to see as big of an effect in that species. And so we know their co-flowering has decreased by about 25%. So what I'm showing here is from the long-term flowering data, the first 10 years of the data set, Potentilla kind of overwhelms Linum, and it's always overlapped. It's always overlapping. However, in the a more recent decade, we can see Linum is starting to kind of edge out in the front here. I think I have it, yeah. And that's because its first flowering date is becoming earlier and it's flowering for more days at the population level, whereas Potentilla's is not. So we're seeing this, they're kind of being pulled apart in time. So, yeah. Yeah.
Like that's almost humanity.
Yeah, that can you comment on the other end of the distribution down here? So yes, there's also, it looks like at the end of the season, you're also getting more overlap. Yeah, and that's because Linum is flowering for a longer time period than it used to. Yeah. So the first question is, do shifts in co-flowering affect interspecific competition for pollinator service between these two species? So we would expect to see less overlap leading to more visits and pollen deposition. And the second question focuses on reproductive success. So do shifts in co-flowering affect reproductive success in Linum? If this is true, we expect to see more fruits and seeds with less overlap. And then finally, the last question is asking about this in the context of climate change, and in this case, drought stress. So could drought stress mediate these effects? It might be that plants are just limited by water and you're not going to see any effect in some abiotic contexts. So we did a field experiment and it was a randomized split plot block design. So we had 1 meter by 1 meter plots, and in the control, we didn't remove any Potentilla flowers. Marie removed half of the Potentilla and then all of them from another treatment. Another question?
Yeah, in an earlier plot with the floristics, you showed 100 species.
Yeah, 60.
What are the other flowers on those plots, or are there none of them? I'm wondering, when you do these pairwise things, the expectations, I think, depend on how many other flowers are in the game at the same.
Yeah, so the question is, what does the rest of the flower community look like in these plots? Because that could affect interactions with other species as well. Oh, I don't know why this is doing things. Sorry. Okay. We'll just leave that there. So these were definitely the most abundant flowers, these two species. There was some geum or some prairie smoke flowering in some of the plots. Trying to think of what else was out there. Some erigeron and aster. But the idea is that, we did a randomized block design. So all of these 3 treatments are grouped together, which you'll see in a slide in a second. So the idea is that we aim to kind of control for any other variation across the meadow where we're doing this by having the plots all grouped together. But by far, these are the most abundant. I might have a picture. We'll see. I can't remember if there's a picture. But then, okay, I'm not sure. I feel like I'm at ESA. Was anyone at ESA this year? The slides were freaking out for a lot of people. So in half the plots, Marie added pollen to the Linum flowers, or in 1/2 of each plot. And so we were able to see if reproduction was limited by pollen receipt or not. Okay, so now here is the spatial arrangement of these things. Oh, I forgot to mention, when she removed the flowers, she removed them in a 4 meter by 4 meter area around each plot. So we had a buffer around that central plot, so there weren't any edge effects. And each plot was separated by 4 meters. And then we also had each group separated by 12 meters, more than 12 meters. So that gives you an idea of how they're arranged. Then half of them received additional water. I don't know, this isn't supposed to happen. Like all these, there we go, there's my water. Okay, so 12 blocks and 36 plots in total. And this was a really dry year. So this was 2018. It was, the snowmelt date was really early and it was very dry. There was very little rainfall. So the plants looked pretty drought stressed, and the watering helped to alleviate that drought stress. So the context of its experiment is set within the idea of a really early year or kind of extreme climatic event. And so what Marie did was she did pollinator observations, recording who visited and how often. We also had an REU student who did a great job helping with those pollinator observations. She collected stigmas and then looked at the proportion of conspecific pollen and then counted the pollen grains as well on the slides. And also she counted seeds from all the fruits that she collected from all the plots. So what did we find? Well, first of all, our water and treatment did increase soil moisture, so that's good. We were able to water, I should mention, with water from a creek that was nearby, and so that was convenient, and she used a watering can and just put it on slowly to let it absorb and not run off. And we did it at the end of the day so that the water wouldn't evaporate. Then we also see that co-flowering overlap did not affect pollinator visitation whatsoever. So the number of visits, I think somehow the slideshow is on. It's like advancing them on its own. So the number of visits per flower didn't change, neither did the number of visitors. And this was also not affected by watering whatsoever. However, for pollen deposition, we found that increasing co-flowering led to a lower proportion of linum pollen on the stigmas, but not a difference in the number of linum pollen grains. So here we're seeing the same number of linum pollen grains regardless. So we call this pollen quality, so that the proportion of conspecific pollen, and so we can say that pollen quality was lower as co-flowering increased. And this is also true whether the plots were watered or not, so across all treatments. However, once we get to the reproductive success variables, we see those were dependent on watering. So fruits that decreased with more co-flowering, but only in the watered plots. So here we've got larger plants up here, smaller plants down here, and they're decreasing as they co-flower more with one another. We see the same thing for the fruit to seed to ovule ratio. So except that it's not affected by size in this case, but in the watering treatment, we see there's fewer seeds per ovule as they're flowering more with each other. Then finally, the last thing is total seeds. And we see that total seeds decreased with increasing co-flowering, and again, only in the watered plots. And here, we're explaining about 33% of the variation in total seed production. So that's better than what we saw before. You might have noticed our square values were pretty low. But for total seeds, this is kind of what we're mostly interested in. And I think the effects of fruits per flower and then seeds per ovule kind of compounded, and we can see that in the total seed production. What about pollen limitation? They weren't pollen limited in any measure of reproductive success. So I know it's a little small, but this is fruit set seed to ovule ratio and total seeds. And then we're showing the difference between the control and the pollen supplemented plants. And it's just, there's a lot of variation and no relationship with co-flowering whatsoever. So the plants where reproductive output seemed to be water limited as opposed to pollen limited. And so Marie made this great schematic to kind of summarize all the results. So we have our two extremes here, 100% overlap and 0 overlap. For pollinator visitation, we saw similar visitation rates between the two treatments, but Marie observed the bees and the flies moving back and forth between linum and potentilla a lot when she was doing her observations. And so we think this is why we saw a lower proportion of conspecific pollen in the areas where they overlapped with potentilla. So here we've got potentilla and linum pollen grains on the stigma, and here it's mostly just linum. And there's been a lot of work that shows that heterospecific pollen can interfere in plant reproduction. So sometimes, most often, I think what happens is that just blocks some of the stigmatic surface. So if you have other species pollen, there's not as much room for your pollen to germinate. We don't know why specifically in our study, but that's something that we see in the literature. And then for seed production, it was increased, but only in the watered plants. So we see this effect of pollination only mattering when they're not totally drought stressed. So again, this shouldn't be layered, but there it is. Okay, if we put this into a broader picture, we see that climate change could have a positive effect on line reproduction via decreases in co-flowering. So as it's getting pulled apart, it's not competing as much for pollination, and it has an increased seed production. But that was only true in the context of not being drought stressed. So any abiotic changes can potentially counteract this effect of dry years. And this is really important because climate change is predicted to increase water stress in Colorado in particular. So in a climate change scenario, in this case, it was really neat that we saw that the biotic and abiotic aspects can interact with one another. And in this case, sorry, it doesn't matter for reproductive success in the linum plants. How much pollen it's getting is limited by water in these really dry years. And I think this is neat because we tend to focus on the biotic and the abiotic effects separately when we're thinking about climate change. And I think there's a lot to be learned by starting to put them together more and see how they might interact with one another. So to summarize, I started out by telling you I was going to talk to you about the ecological consequences of different flowering times under climate change. We saw that climate change can lead to a decreased population growth rates in plant species, but not actually because of this earlier flowering and frost damage. So despite the fact that this was a really big effect and really visible and something that seems like it might matter for the populations, it didn't as much as reductions in survival. And again, this points to the importance of considering the whole plant life cycle when you're thinking about population level effects. Then we also saw kind of an unexpected effect, consequence of earlier flowering with warmer and drier soils, potentially mediated through this albedo pathway. And then finally, the changes in co-flowering due to climate change can alter plant reproductive success, but that's going to depend on the abiotic environment as well. So with that, I'd like to thank my students, undergraduate students, master's students, collaborators, funding sources, and I'm happy to take more questions.
So if we could have a grad student start with the questions or a postdoc, that'd be great.
Yeah.
What does the research show about early flowering in annuals? Is that well established versus perennials in terms of like reproduction versus survival being important in those two different life history strategies?
Yeah, that's a good question. So the question was, what do we know about annual plants and earlier flowering under climate change and their population dynamics? So certainly for annual plants, we do know that their population growth rates do tend to be much more sensitive to perturbations in reproduction, as you might guess, because it's an annual plant, right? There's one shot at reproducing. And they can't reallocate resources across years. So we do see that. And so you would expect to see a much bigger effect in an annual plant species. And so in Colorado, the community is vastly dominated by long-lived perennials. So that's why we're working on them. It's hard to find annuals to work on. There's a biennial, a mustard, that I've done a little bit of work on with a collaborator, and Jill Anderson did a really amazing quantitative genetics project and found that earlier flowering in that species was mostly driven by plasticity in addition to some evolution, some selection for earlier flowering, but also a lot of plasticity, as you might expect. Yeah. Yep. Yeah.
So this abiotic and biotic factors interacting is pretty cool. I was just wondering about another biotic interaction, which is, so you talked about competition between, I was wondering about herbivory, and is that, you think, because you mentioned that there is herbivory in the first part of the talk, so do you think that changes?
Yeah. Yeah, so the question basically is, what about other biotic interactions? Obviously, I just focused on pollination, but what about herbivory? There is some kind of beetle, I think, that can eat linum flowers. I don't know what it is, but I can't really speak to how it might affect that study because we didn't measure it, and I don't think we saw a ton of it. We did see there is also on Linum rust, a fungal rust that happens on it, and most of them had the rust, like most of the plants in this study did, and we don't know much about that, and I think that's a really interesting one to do some more work on too. So yes, I think other interactions are very important, and ultimately it'd be great to focus on more than just one biotic interaction. Yeah. In the back, I think you first.
Yeah. With the early snowmelt, how often does that happen? Is it like becoming like a common thing every year, or is it like once every 5 or 10 years that you have this like
early snowmelt? Yeah, how often is the snowmelt becoming earlier? So there is a trend toward earlier snowmelts, although there's a lot of variability around it. So, for example, in the 45 years or so that snowmelt has been measured at the field station, it's getting we've seen about 2 weeks earlier on average with some substantial variation. Does that answer your question? Yeah, cool. I think you're next and then Nick, yeah, in the back.
For the first part, where you had 3 different populations and your varietal differences, how different are the populations from each other?
Yeah, how different are the populations from the first part of the talk? Yeah, you might note that I didn't talk anything about elevation and how it might affect the populations because we only have one population at each elevation, and they are different. So the lower elevation, there's more grass, I would say, in the plant community. That population's not super different from the mid elevation, but when we get up to the higher elevation population, that's on a steeper slope, and the plants are not as dense. And so I think the soils are shallower too, and that might be one reason why we see early snowmelt having a negative effect on vegetative growth in that population and not the others.
Do you know if there's genetic differences?
I don't know if there's genetic differences. Yeah. They're pretty far apart from one another spatially. I don't even want to try to guess how far apart they are, but they're far. Yeah. Nick.
So a working model in conservation, as somebody who thinks especially about connectivity, is that we conserve connecting landscapes from south to north or from downhill to uphill.
Mm-hmm.
And what I'm taking from your talk is that this is like the disastrous working model of conservation, because if you connect uphill, well, uphill is death. And so like for how what types or range of species is the sort of uphill death failed conservation strategy?
Oh, that's a hard question to answer. Yeah, so Nick's question was, relating to habitat connectivity. We're trying to often connect populations north to south or uphill to downhill, and the uphill ones, if they're all dying, maybe that's not such a great model. So how often is that a thing? How often are the yeah, for other species? I don't think we have enough information to give you a very good answer on that. But I think the positive news is, we do see species certainly expanding their ranges up, but how are those populations doing, and are they just there temporarily? Can they stay long term? I think is a really important research question to answer. Yeah. Mm-hmm.
I was wondering if you noticed any specialist pollinators of the species, or do they mostly share?
Yeah, the question is, are there any specialist pollinators? So the sunflower, the potentilla, and the linum are all very generalized, especially the linum and potentilla. There's about half the visitors are flies and half are bees, and a variety of some bumblebees, but mostly solitary bees. and lots of syrphid flies and bombyliids. Oh, good questions. Is that it? I think. Thank you. Thank you so much.